In this video, we're going over the experimental procedure for the qualitative analysis of carbohydrates, which is lab 12. For this lab, we have a series of tests that need to be performed. From the drop-down menu on the left side of the simulator, we're going to specifically do the Moelisch test, the Fellings test, the Benedicts test, the Tollens test, and the iodine test. In each of these tests, we are going to utilize the same 4 samples of carbohydrate, meaning that we're always going to be testing the monosaccharide glucose, which is in a test tube labeled letter A, the disaccharide lactose, which is always in test tube B, the disaccharide sucrose, which is always in test tube C, and lastly, starch, which is a polysaccharide that is always placed in test tube D. So the first thing that we're going to do is select the Moelisch test. In the Moelisch test, remember that this is utilized to identify any carbohydrate in a sample. It doesn't matter if it's a monosaccharide, disaccharide, or a polysaccharide, and it's going to be given by that purple ring that forms. To do the experiment, what we have to do is place some of the Moelisch reagent on each of the test tubes. Next, we are going to grab the test tube, put it by the 2 containers, and now place some concentrated sulfuric acid on the test tube. And we're going to do this for every single sample. Then you are going to select the information that is here by the letter I that is in red and in that yellow circle. And you can see that this is the explanation of the observations that we see in the Moelisch test. You can click on that I icon several times to copy down all of the information. Let's proceed now to look at the experiment for the Fellings test. In the Fellings test, what we are looking at is the creation of a reducing sugar. What that means is that that sugar, that it is a reducing sugar, is going to reduce the reagent. In this case, this is a copper reagent, so it's going to make a copper 1 product. We have the same 4 samples, and in here we have 2 containers. We have the blue Fellings solution and the clear colorless Fellings solution. To each of the test tubes, you're going to put some of the blue Fellings solution. Followed by the clear colorless Fellings solution. As you can see, each of the samples now have a deeper blue color. Now the samples are going to put in a water bath. If we were doing this in the laboratory, we need to boil our samples for at least 20 minutes. But you can see here that the creation of that orange color product represents the 2 samples that contain reducing sugars. And by clicking the I icon, we can see that specifically glucose and lactose are the reducing sugars. Now we're going to proceed to look at the Benedicts test. In the Benedicts test, similar to the Fellings test, we are going to be studying reducing sugars. So since the observations of the Fellings test is that glucose and lactose are our reducing sugar, understand that we're going to see similar observations here. In this experiment, we are going to place some of the Benedicts reagent to each test tube. Now we proceed to put the test tubes in a water bath. And similarly, if we were doing this in the laboratory, we have to wait 20 minutes for boiling the samples. And as you can see, similar to what we saw in the Fellings test, because this is another test for looking at reducing sugars, we can see that glucose and lactose are our reducing sugars because they are reducing the copper 2 in our Benedicts reagent into copper 1. The tolerance test is also a test for reducing sugars. But here we're going to observe that instead of creating a copper compound, we're going to create a precipitation of silver. So in order to do this experiment, we're going to take that tolerance reagent and place it on each test tube. Now the samples need to be placed in a water bath. And now by clicking the eye icon, you can see that indeed the reducing sugars that we have here in the carbohydrates that we tested are glucose and lactose. And in the tolerance test, that is represented by the precipitation of that reduced silver. The last test is the Iodine Solutions test. This test is specifically for detection of starch in a sample. So we're going to put some of the iodine solution on each of the test tubes. And the one that contains starch is going to turn blue. When we do this in the laboratory, it turns into a deep, deep blue sample. It's almost black. But you can see here in this computer simulator that the only sample that has starch is the one in the letter D. And it's because iodine, as you can see, is going to form a complex with iodine, and that's why we have the creation of that blue color.